Can antibody titers predict clinical influenza symptoms or who might suffer a worse course of disease? That's what we're going to talk about on Microbial Minutes. This is ASM's update on what's hot in the microbial sciences, the Correlates of Infection edition. I'm Julie Wolf, and we'll be discussing a report from ASM's MBio journal that shows that differential gene expression can predict influenza contagiousness and illness severity in people. So we know that influenza remains a serious threat. Just in the 2017-2018 influenza states alone, 83,000 people died due to complications resulting from their influenza infection. Now, better prognostic markers might help doctors or healthcare workers to better care for those who are at risk for a worse course of disease, or perhaps be able to stem transmission if they know that someone is shedding viral particles despite not showing any symptoms. Because influenza is seasonal and something that most of us have experienced once, if not more times in our lives, we're all carrying around various antibodies to different influenza viral parts. Most of those are to either the neuraminidase or the hemagglutinin, the Na and the Ha, that give different flu strains their name. As shown on the electron micrograph to the right-hand side, you can see the little spiky parts that are coming out from the circular influenza particles. Those are the HA and the Na particles. That in particular is an H1N1 influenza strain. And so what the authors of this study wanted to ask is, can we use those pre-existing titers that we all carry around with us to predict patient outcome during influenza outbreaks? What they did to answer this question is to take a trial of 83 human volunteers, people who volunteered to become infected with the 2009 H1N1 influenza A virus. During the study, their blood was drawn, and the global gene expression pattern within the peripheral blood leukocytes, those are the white blood cells, were investigated both before infection, so the pre-symptomatic, during infection, during the entire course of disease at various time points, and then as people recovered during the convalescent period as well. These gene expression patterns were then correlated to both the patient's symptoms, viral shedding to indicate contagiousness, and to those pre-existing serum antibody titers. Now, on the next slide, we'll see that one of the first things that they found was that the baseline peripheral blood leukocyte, or PBLs, the gene expression did not correlate to clinical income, which means that we can't predict outcome prior to infection very easily. However, very early on during infection, a viral shedding associated gene signature was identified in most people who have virus that is present in their nasal wash as early as 2 days post-infection, which is before many people are starting to show influenza symptoms. 3 different groups with various levels of responses were identified. We can see those in the upper graph on the right-hand side, where various levels are color-coded here in blue for not strong response, the yellow for intermediate, and red for strong response. The viral shedding associated gene expression patterns were then correlated with the duration of clinical symptoms, how long someone suffered from influenza, the length of viral shedding, and pre-existing antibody titers. So the pre-existing antibody titers did correlate with the level of response. However, only the antibody that was to the globular head of the HA, not to the stalk. So if we recall how the HA looks as it comes out of the viral envelope, there's the you can consider it kind of like the tree branches part is the globular part, and then there's this trunk-like stalk. And only the antibodies that were to that globular head really were statistically significant between the people who had no response and the people who had variable responses. So if you look at those lower graphs, the F, G, and H labeled on the right-hand side, you can see that the only place that there was a significant difference between people who had that intermediate response and no response was when they looked at those HA antibody titers to that globular head. That makes sense because that's the part of the viral envelope that we interact with the most. However, that also varies over time with different strains, with different hemagglutinin genes, and with different mutations within the genes themselves. So they additionally identified other populations of genes that could correlate with the length of shedding and illness severity. This is not the first time that scientists have tried to investigate correlates of infection for influenza virus. On the next slide, we'll see that there are a number of different studies that have been conducted to identify different genes or aspects of the patient that may predict who is going to have a worse course of disease. Some of these have even identified potential hits, but those were identified in an H3N2 virus, and it's important to ensure that any correlate of infection is going to be applicable to all the different influenza strains that people can suffer from. Now, ASM has many different influenza resources, including a look at the ongoing clinical trial, which involves looking at antibodies against that stalk region of the HA, and as well as a summary of what current point of care testing for influenza is, which this particular study and these other correlates studies are really looking for ways that we can identify point of care tests that will indicate who is going to be at risk for a bad course of infection. So today we've heard that gene expression patterns can predict influenza shedding and disease severity, but whether that is going to translate into a test immediately, probably not, because it's unlikely. To keep up with the latest research in this area, however, go ahead. I'll be with you next time on Microbial Minutes.